Thanks, Mike. There's our disclaimer statement. You were right. Okay, so we're an AIM-listed pure-play biotech company. So we follow the old-fashioned biotech model of raising money from investors, hopefully deploying those funds sensibly against research and clinical stage programs to deliver a return in due course if the data goes our way. We're working with very differentiated stem cell technologies, and I'll try and describe them as best I can in a moment. This is not a science presentation. I'm not a scientist myself, which kind of helps working in a business like this. But there are plenty of boffins around, as you might imagine, in the business. We are focused, though, on very significant disease conditions. Largely, these are diseases which are characterized by not having any available treatments currently available for them. And we are playing into an increasing demographic, by which I mean an aging population. So most of the things we're developing are chronic diseases which are largely associated with an aging population in the developed world. We do have some fast track and orphan designation for our retinal program, and I'll come back to that specifically in a few moments as well. All of our programs now are in the clinic, so we are an out-and-out clinical stage business, which is good, and I'll explain why that is in a moment when it comes to getting a return back for our investors. If there's time towards the end, I'll tell you a little bit about an emerging platform that we're also developing now in its research phase, which is called exosomes, and these exosomes are actually derived from the stem cell-based platforms that we've been developing for the last 15 years. So it's kind of ReNeuron Mark II, but we have a lot of hopes for that particular platform, and I'll explain why and show you a little bit of data if there's time towards the end. We're currently capped at just under $100 million, I think probably about 90 million actually as of today. As you can see in the second bit of the slide, a large part of that valuation is actually accounted for by cash on the balance sheet. That's very important for a company like us because we have a very expensive and very important job to do over the next 3 years, and we now have the money to be able to do it. And that is to generate data in clinical trials in 2 of our core programs to get them right the way through to market application. So it's fair to say in the past we've been a serial fundraiser. We've managed to get by, but now we have the funding and the investor base to really get the job done. And there is no cheap way of doing late-stage clinical trials. They involve a large number of patients, and they cannot be done cheaply if you want to design them in a way that will give you unequivocal data. Okay, so we're not going to shortcut and try and do things, you know, on the cheap. We've got to do well-designed clinical trials that will garner unequivocal data. And that's important because ultimately these programs are going to be partnered or sold to large pharma or biotech companies. And the previous presentation showed you why that will happen if the data is positive, because pharma companies can shortcut those very expensive and very long development timelines by buying programs like this. That is why the biotech industry exists, to fill the paranoid pharma pipelines that currently exist. Pharma companies are not particularly good at innovating. They look to biotech companies, especially those involved increasingly in novel therapeutics, immunotherapy, gene therapy and cell therapy, to fill pipelines. And so it's very important that we're able to garner data that's unequivocal in their eyes. They can see that it's worth an investment, that we've lowered the risk for them, and they're going to pay for that. And quite simply, they can afford to pay a king's ransom for the best clinical stage assets in the business. So again, it's important for us not to have to run and chase to get deals to fill the coffers. It's more important for us to be able to run our own trials, generate the data, and then see what value we can get attributed to them through deal making once we have those data in hand. Hopefully, 1 or 2 of those investors will be familiar to you. Why are we involved in stem cells? Well, we are a pioneer in this technology. We've been around for 15 years. It takes time to develop the data sets you need to make progress. But we've always been strong believers in the notion of stem cell science to address very intractable diseases, to try and get at the underlying causes of diseases rather than merely acting on the symptoms. And we've demonstrated, I think, through a large range of clinical data and preclinical data over a large number of years that there is real potential in this technology. The big challenge is how do you convert it into products? something that can be manufactured, expanded at scale, sold at a cost of goods that's going to be interesting to a pharma partner and, of course, interesting to a healthcare payer. You have to tick both of those boxes, quite apart from having great clinical data and exciting science. There's got to be a commercial reason for doing this at the end of the day as well. So the two technologies that we've been working on for a large number of years are sort of represented diagrammatically here. And again, I'm not going to take you through the science here. We have two products. One is called CTX. It's a neural stem cell line originally derived from brain tissue. And we also have what's called an HRPC cell line. That stands for human retinal progenitor cells sourced from retinal tissue. The CTX cell line is being used in our stroke program and our limb ischemia program, and the retinal cell line is being used in our retinitis pigmentosa program, which is a retinal degenerative diseases. So obviously we're targeting the cell lines really back to the organ of origin, the exception being limb ischemia, which is actually a condition in the lower limb, and we're actually using a brain cell to address that particular condition, which sounds a bit odd, but I'll explain why when we get there. So what is CTX? What's the product we've developed? Well, CTX, as I said, it's a neural cell line, but we've developed and patented a way to expand the cells, first of all, by genetic modification, then to cryopreserve them using a patented formulation that enables us to do so. And that's important because it means we can manufacture these cells at scale under GMP, good manufacturing practice conditions, and then cryopreserve them, stick them in a cryoshipper, which is that funny little bottle thing you see on the left-hand side. And those cryoshippers can be shipped around the world, stored at clinical sites, and the cells can be then thawed, diluted if necessary, and administered on-site, on demand, when the patient requires it. And by the way, these cells can be administered to any patient that presents with the diseases concerned. These are what are known as allogeneic cells. They don't require, or our treatments don't require cells, stem cells from the patients themselves. This is a cell line that was selected for its immune characteristics, and it does not engender an immune response in patients, even though the cells are not the patient's own cells. And we know this because in all of the clinical trials we're doing, in the brain, in stroke, in limb ischemia, and in the retina, in neither of those three cases are we immunosuppressing the patients. And of course, immunosuppression is not a good thing for patients. It's a safety risk in and of itself. So if we don't have to do that, that's a really good thing clinically for the patient. And we don't need to do it either with CTX or with our retinal cell line. So that's broadly what the product is. Here's the pipeline, your classic biotech company pipeline. You've got to have one of these. So here's ours. 3 programs. As you can see, 2 are using the CTX cell line. 1 is using the retinal cell line. And our exosome program is listed as well. It's a bit more speculative in its research phase, but it's on there because it's important to us now. White is what we've already done. Blue is where we are currently at with each of these programs. And green is where we're looking to take these programs with the money we already have in the bank. So as you can see, in the case of stroke and retinitis pigmentosa, we have enough money now to take those programs right the way through pivotal clinical trials to the point of marketing application. With CLI, we can take that program through phase 2. We will need to do a phase 3 study if the phase 2 data plays out, and that actually isn't in the use of proceeds. But if we can make success with the other programs, that's not going to be a big issue for us, especially with stroke, because it's the same product we're using for CLI. So the better the data we can garner with stroke, the more confident our investors, I think, will be in the limb ischemia program as and when we want to take that one forward. And of course, it may not be an investor, it could be a pharma partner that does so. Okay, so now quickly on the programs in turn, starting with the retinal program. Why am I starting with that one? Well, it's one really where the news flow has been most recent, and it's important to us primarily actually because it's our first clinical endeavour in the US. Now, we are a UK-based company. We've done all of our clinical trials thus far in the UK, and quite a bit of that work's actually been done up here in Scotland, in Dundee, Glasgow and Edinburgh. But it's important that we look beyond that now as we mature, and this is the first program where we're taking it directly into the US for its first clinical trial. The idea of these cells, the HRPCs, is to essentially rescue lost photoreceptors. So photoreceptors are the rods and cones that exist in the retina that give us vision. And in these retinal degenerative diseases, essentially you lose those photoreceptors. So we're looking for these HRPCs to engraft and then differentiate into those lost photoreceptors, either to arrest deterioration in these patients or, best case, to actually restore a modicum of vision. And it's amazing if you actually talk to these patients, even a very, very modest improvement in their ability, even just to sense shadows where they couldn't before, makes a huge difference to their lives. So this is not a panacea. This is not sort of full restoration of vision. That would be remarkable. But some modicum of improvement would be transformational for these patients. And of course, it would make the treatment extremely valuable commercially as well. So we've been working with a lot of smart people over the years to develop this. It started out of Harvard. That's where we licensed the original technology from. We've been working with Moorfields in London. The Foundation Fighting Blindness is the US's largest charitable foundation that looks at blindness more generally. And they've helped us with regulatory pathways, and they've actually put a little bit of money into the early stages of this program as well. The first target we're working on is retinitis pigmentosa, as I mentioned. This is an inherited degenerative disease of the retina. What you get is what you see in the image on the right there, this gradual loss of peripheral vision, ultimately leading to blindness. And that can happen over a number of years. The important thing with this particular program is we have two designations for it. One is orphan drug designation in both Europe and the US, and fast track designation by the US FDA, the regulatory body in the US. Orphan drug designation is very important. You get it if you're really addressing rarer or smaller patient populations. And what happens there is if there is no better treatment available at the time we get this to market, then we will have market exclusivity for a period of 7 or 10 years, depending on whether it's Europe or the US, for this treatment. So there are plenty of orphan drugs out there that have this designation, and it's kind of like a race, I guess. We've got to generate clinical data that at the point of application really means there's no other better treatment available. We'll get that market exclusivity. And, we've got a good chance of doing that, providing we can generate the data, because RP is a completely unmet medical need right now. So, it's important that we target these very intractable diseases because there is a prize to be had at the other end. Competition is not something we are over-encumbered by in this particular field. Fast-track designation means that the FDA will really do their best to make the regulatory pathway go much faster than it otherwise would. So it gives you certain advantages when dealing with the regulator, or the FDA specifically in this case. So we have started a phase 1/2 study, a combined safety efficacy study in the US. We're about to treat our first patient. All being well, if the data pans out, that will lead us into a pivotal phase 2/3 study at some point next year, next calendar year. And that is it, 2 clinical trials to market. Again, that's what a program targeting rare diseases in relatively small patient populations will get you. It's a much swifter regulatory pathway. Pharma companies love that kind of stuff these days because if they can buy into enough of these, it very rapidly builds a revenue stream with programs that are much swifter to market than the kind of 14-year, $2.5 billion trek that you saw in the previous presentation. So that's why this is becoming a much more investable program now. Okay, so back to stroke, which is the program I guess we're best known for as a company. Why the hell are we addressing stroke? It's a graveyard indication in the parlance. Virtually everything that's been tried in stroke has failed. Well, you have to make some distinctions. And what we're really targeting at is what you see in the headline on the slide here. It's the consequences of stroke. So basically, if you survive a stroke, and you've got about a 2/3 chance of surviving a stroke if you have one, and if you do survive it, you've got about a 50-50 chance of being left disabled by it, either cognitively or by way of motor deficit. And this treatment is targeting the motor deficits. Andrew Marr would be a classic candidate. He isn't a candidate for our clinical development plans, I hasten to add. If it all went horribly wrong, that wouldn't be good PR, of course, but it is a good example of what a stroke patient will go on and suffer. It's this unilateral deficit you have on one side of the body, this hemiparesis, this inability to move on one side of the body, especially the upper limb. You can kind of compensate for lower limb disability by, swiveling your hip or limping, but it's very difficult to compensate for upper limb disability. So we're hoping that CTX will engender a degree of deficit improvement, or I suppose reduction, in these patients sufficient to make a meaningful difference in these patients' lives and therefore to make them less reliant on health and social care. And that is where the pharmacoeconomic argument is for this treatment. No current treatments available for stroke survivors other than sporadic rehabilitation or physiotherapy. The only marketed treatment for stroke is alteplase, which is essentially an antithrombolytic in the acute phase. You have to get to the hospital within 3 or 4 hours to have it administered, which means that about 93 or 4% of patients render themselves ineligible for that one treatment. They just don't get to the hospital in time. We've published very extensively over the years across all of our programs, actually, preclinically. So whenever we generate data, we publish it. We're quite happy to have our work exposed to peer review and scrutiny, and we actually do that when we apply for grants as well, and we've been successful in garnering quite a number of grants over the years. And again, that exposes your programs to peer review, scientific scrutiny in a competitive environment, and to our minds, that's a good thing. And we know a lot about how these cells work, and they work in a number of different ways, and I won't bore you with the precise mechanisms of action. If you want to read the published work, then I'll send you the papers, and good luck with that. Actually, they're very interesting if you can sort of cut through the jargon. So where are we with this program? Well, we've already finished a phase 1 study in 11 disabled stroke patients, so we were treating stroke survivors from the get-go. It's a simple neurosurgical procedure. The patient will typically stay in 1 or 2 nights and then goes home. We were very encouraged by the data we saw from this study. It was done up here in Scotland, in Glasgow, under Keith Muir at Southern General down the way. No cell-related or immune-related adverse events, and encouraging evidence of efficacy, which led us into a phase 2 study, which is currently ongoing again here in the UK across 10 centres up and down the UK. And the important addition in this particular phase 2 study is what you see in the panel on the bottom right there, if you can see it. The primary endpoint measures the patient's ability to perform a useful task with the affected side of the body. So as you can see, the patient is attempting to lift a wooden block from desk height onto a platform. That's a very objective primary endpoint for a clinical study of this nature. Very meaningful from the patient and a clear one to read. So we're not looking for a nuanced change here. We're looking for perhaps a relatively small number of responders, a meaningful clinical benefit that's going to be meaningful for the patient and, as I mentioned, meaningful ultimately for a payer if we can get this through to market. So the phase 2 data will read out in the middle part of this year, and all being well will lead to a pivotal phase 2/3 study, which we're planning now, and that is very likely to be a combined US-European study, which we'd hope to commence at the back end of this calendar year. Just moving on to critical limb ischemia, very briefly, what is CLI? Essentially, it's a loss of blood flow to the lower limbs. Very common side effect of diabetes, unfortunately. Around 70% of CLI patients will be diabetics. Very large healthcare burden, again, it's a classic bed blocker. No treatments available other than surgical revascularization. Most patients won't be eligible for it because of their wound healing problems, because of their diabetes. So we're looking for a cell therapy treatment here, administered into the calf muscle, essentially to revascularize, to grow the microvasculature. And we've seen this very, very clearly in the animal work that we've done. And that's because our CTX cells, those brain cells, why are we putting them in the leg? Well, they are proangiogenic. They are highly predisposed to generate new blood or promote new blood vessel growth, which is precisely what's required in this particular disease condition to prevent what you see ultimately in the image there, that's dry gangrene, which will ultimately lead to amputation. So we have a phase 1 study ongoing at the moment, again up here in Scotland, this time in Aberdeen, and all being well, that will lead straight into a phase 2 study, which again we're planning to commence in the second half of this year. So finally, just a quick word on the nanomedicine platform. I've got 2 minutes left, so I should be able to do it. What the hell are exosomes? Well, exosomes are particles that stem cells secrete, and they secrete them because these exosomes then send signals to neighbouring cells. So it was previously thought that exosomes were really vehicles to get rid of cellular junk, but actually they play a vital role in signalling to neighbouring cells to engender certain processes. And we believe that exosomes are partly, or perhaps mostly, responsible for giving our CTX cells their beneficial effects. But we also believe they could be a therapeutic agent in their own right. And why do we think that? Well, without boring you as to why we did this, but we looked at whether we could target exosomes against cancer. That was based on a bit of early work that seemed to suggest they might have anti-cancer properties in certain settings. So here's just a quick snapshot of some very early and speculative work we did using a glioblastoma model of cancer in a dish. So this is essentially a tumour cell, or a tumour rather, that you grow in a dish, a glioblastoma, brain cancer tumour. And what you see in the photographs on the right-hand side, the left-hand column of 3 there is an untreated tumour growing in a dish. There's an active control, which is a P13 kinase inhibitor. As you can see with the red line on the graph, if you continue to administer that drug, you can suppress the tumour growth. As soon as you stop administering, tumour growth starts again. And then with the exosomes, we didn't really see much for the first 2 or 3 weeks. It tracked the control. But then all of a sudden, the tumours collapsed, and all we got in the dish was cellular debris. So clearly something seemed to be happening here. First of all, we thought it was a mistake, of course, because the graph looks like it's a mistake. But that blue line is actually the mean of 5 experiments. So there is clearly something going on here. We then replicated that, this time in an in vivo environment, so in an animal model. And the positive control this time was temozolomide, which is the only marketed treatment currently for glioblastoma. And again, without going through the graph in detail, similar pattern. Untreated control just keeps growing. The drug-treated positive control, the drug-treated tumour, you can suppress it. But with the exosomes, not much happens for a few weeks, and then suddenly you get this massive reduction in tumour size, and full regression actually in the case of 2 of those animals. So what do we think is going on here? What we believe may be happening is that the exosomes are programming those neighbouring cancer cells to adopt a normal process of cell cycle death, apoptosis in other words. And cancer cells are really great at doing 2 things. First thing they're great at doing is hiding themselves from the immune system, and that's why the emerging field of immunotherapy now exists. The second thing they're really good at doing is not dying. So we believe that this may be a second modality against cancer to reprogram those cells, essentially to die, to behave more like normal cells. So again, very speculative, very early, certainly nothing in the share price for this right now, but we hope could give us a really good second platform, or third actually, string to our bow in the future. Just in the interest of time, so I'm into my question time already. Here's the pipeline, also not the pipeline, the milestone chart over the next 3 years. So all of these boxes represent meaningful clinical data events. They start in the first half of this year, as you can see, with stroke phase 2 in the top left, and continue over the next 3 years, for which we have funding already, leading to pivotal data in 2 of those programs, phase 2 data in CLI, and perhaps more speculatively, all being well, some clinical data with our exosome, our first exosome candidate as well, which of course is very likely to be a cancer indication, but we haven't announced which one yet. A quick nod to the management team. We've really built out our management team now to cover all the key functions that a good biotech company should have, medical, regulatory, clinical, and so on, and some very experienced non-execs as well who've worked in the industry for many, many years, all that good stuff. So just to wrap up then, the investment proposition, as we see it, we have highly differentiated stem cell technology. These are scalable and cost-effective stem cell technologies, a way of generating something that is more akin to a pharmaceutical product than just interesting science. We have a great competitive position, very high barriers to entry, as I guess is self-evident. We're addressing very significant diseases, and we are very much a clinical stage company now with a clear shot on goal, if you like, across those programs with the money that we have. And therefore, based on the current share price and valuation, significant opportunity in our eyes to build value for our investors over that 3-year haul. And with that, I'll say thank you.
Thank you, Michael. Questions, anybody? Ooh, gentleman here. Okay, what is your market cap currently?
It's about 90 to 95, I think, when I last
Certainly
looked.
just above cash, really.
Yeah, not far above cash. Yeah.
Hello, can I ask you about how you would monetize your successful treatments? Would you sell them, keep them, JV them, or royalties?
What we'd like to do is be funded sufficiently to be able to take these through to market, should we so wish. And that's not an impossible prospect when you think about it. These are highly specialised hospital products. So even in the US, you would not need a large sales force to market these to neurosurgeons, in the case of stroke, for instance. So I think the position we would like to get ourselves into is to have money to be able to launch products in key markets, and then have our conversations with pharma to say, well, we could partner with you, we could sell the business to you, but we don't need to. And that's the way high-value pharma deals are done. So you don't want to go chasing deals. And of course, the data has to be good. Data is everything here. So we have to ensure that we've got as I said, unequivocal data through well-designed clinical trials. But we're keeping our options open. It could be, it's probably going to be a partnering deal of some description. It could be a wholesale takeout, because bear in mind we have platforms here as well as just single programs. But I think we need to be in a position to keep our options open, and you can derive higher value deals by doing that, and there is plenty of precedent actually in our field for that. So that's the broad strategy, but it's not nailed down in stone. We have to generate the data first.
Is that more likely then beyond phase 2 into phase 3?
Yeah.
That's likely to happen?
Yeah, I think it's very unlikely that we'll partner any earlier than phase 3 on the vascular programs. Conceivably off phase 2 in the retinal program, that's rather more investable at an earlier stage for a number of reasons. But again, I think the important thing is to ensure that we can really get the job done. And certainly we know that some of our major investors, and I won't say who, have expressed a willingness to reinvest when we do get to the point of market application for just those reasons.
Well, Mr. Woodford again, was it?
I couldn't possibly comment.
It was. Clearly was then. Maybe not. Any other questions? Oh, we'll have time for 2 more. We'll just quick questions.
Time up.
So one at the back first.
Can I just ask you a broad investment question? You know these unique platforms you've got, they're patent protected.
Yeah. Yeah, they're all, I mean, we've got a very, very broad patent estate, I mean, because we've been doing this so long. Yeah. So, as an example, the CTX cell line is a patented entity in its own right, and those patents have issued. The process of expanding the stem cells is patented, the cryopreservation technology is patented, the retinal cell line is patented. So it's multi-layered, and of course we have a lot of trade secret coverage as well in terms of processes. My question is, because I'm a field science and all that stuff as well, but see your competitors, surely that doesn't preclude them from actually developing their own positions in these markets? Yeah, no, but think about it. We're dealing here, so think for a moment about the concept of a generic cell therapy. These are living cell products. Every cell is unique. Every cell line is unique. Bear in mind that CTX wasn't just developed as the first hit. It was selected from over 120 clones back in 2002. So sure, you get a new entrant coming in saying, let's copy CTX. They would have to, first of all, try and clone it using technology that we patent. They then end up with a cell line that may exhibit similar characteristics, but wouldn't be identical. And then for that reason, they would then have to take it through a full development pathway, the same as we've had. These are not synthesized chemical drugs that you can just copy using a clever chemist in a lab. They are living cell products. So patents are important, especially over processes and manufacturing especially. But the actual concept of generic approaches to this is to my mind, a non sequitur. The final part, this is the money question. See the £72 million you've got. How long would it take to burn that kind of money on the basis? Over that 3-year haul. So we expect that cash to last out to the end of calendar 18. That's a lot of money. So we're going to be spending a lot of money over the next 3 years. And as I said at the start, there is no cheap way of generating phase 3 data. You have to do it the way that potential pharma partners will want to see it done. And, there's just no easy way around that.
One more question just down here.
Just a quick, maybe I didn't listen well enough, but I'm assuming you're at the pre-IPO stage, yes?
We are a public company on AIM, yes. Sorry,
you're a public company on AIM?
Yeah,
okay.
yeah.
Thank you very much, Mike.
Thank you.
Thank you.